All right, so to continue with the HIV virus, all right, I've got a little diagram here. Let me put up here just so that you remember that we're talking about HIV here. I've got a diagram here showing a eukaryotic cell. This is our HIV virus, all right, and in terms of gaining entry into that eukaryotic cell, that envelope around the outside of the HIV virus is going to make contact with receptors that are on the outside of the eukaryotic cell, all right? Now, in terms of that, typically when we talk about receptors, we tell you in biology class that receptors are very specific for ligands, all right? The truth is there's a little bit of wiggle room there. And if a protein is close, it can interact with a particular receptor. And that's what's happening here. The HIV virus has evolved to use something we already have on the outside of our cells in order to gain entry, okay? So the receptor on the outside of our cells that allows entry is called CD4, all right? There are tons of different receptors on the outside of immune cells, all right? The CD stands for cluster of differentiation, and when we get to the immune system, I'll talk a little bit more about these. All I want you to know, CD4 is what's going to be, in part, what allows HIV in. It also needs a co-receptor, all right? So the co-receptor and the CD4 are both needed in order for that HIV virus to latch onto the outside of the cell and to fuse with the cell membrane so that it can deposit that viral material into that immune cell, okay? So in terms of our entry here, this should look a little bit familiar when I was talking about the generic absorption, all right, the fusion of the virus with the cell, the uncoating of the virus to release, in this case, the RNA, all right, and we'd also release in there that reverse transcriptase as well, okay? In some cases, that virus can move to the nucleus and it can integrate into the genetic material, so it actually becomes part of the cell itself, all right? It doesn't necessarily have to integrate. It can continue to replicate and make more viral particles without integrating, okay? But the integration step is what allows that HIV virus to remain latent in some people, okay? In terms of that, after we've got the encoding, then we're going to have the synthesis. So we're going to have transcription and translation of the viral components. We're going to assemble those components together, all right? And in terms of that, then we're going to have the release or the budding of the virus off of that eukaryotic immune cell, okay? And you're probably going, well, yeah, okay, great, Professor Riley. You already went through this whole process in another video, okay? The reason I put this up here again is because now this is going to represent places where various inhibitors are going to work, okay? So one type of inhibitor that can interfere with the process of replication of the HIV virus is going to be a reverse transcriptase inhibitor, okay? So that enzyme right here that comes in, the inhibitor of a reverse transcriptase is going to make it so that that particular enzyme won't work. If that virus, the HIV virus, can't make a DNA copy of that RNA virus, you're not going to get replication within the cell, okay? And in terms of the inhibitor, you may have heard of AZT is going to be an example of a reverse transcriptase inhibitor, as far as HIV treatment. And it does cut down on viral replication to kind of keep the viral load lower, all right? If the viral load is lower, you aren't going to be losing as many immune cells, and the person isn't going to suffer from the symptoms associated with AIDS, okay? So reverse transcriptase is one target we can hit, okay? Another target we can hit are going to be protease inhibitors, okay? One thing I haven't mentioned yet about HIV virus, okay? Proteases, based on their name, proteases essentially will cut proteins, okay? Now in the process of synthesis, once those proteins are translated, once those viral proteins are translated, in order for the HIV virus to be functional, some of those proteins need to be trimmed. And the way they're trimmed are going to be with proteases, okay? So I talked about the reverse transcriptase coming in. You also have some proteases that are going to be brought in with the HIV virus as well, okay? Because those proteases specifically trim those viral proteins so that capsid and all of those enzymes can be put together in a nice little package, okay? So protease inhibitors are another way to target the HIV virus. I'm not so worried that you know the names of protease inhibitors, okay? What I want you to focus on, because I gave you AZT here, because that's one you've probably heard of. You probably haven't heard of the protease inhibitors, and that's okay. What I want you to concentrate on are where in the process of replication you're going to inhibit things. So reverse transcriptase, the protease. The other one that's going to inhibit things is going to be with the integrase, okay? So targeting this step here. If that viral DNA isn't allowed to incorporate into the eukaryotic cell's genome, it's not going to be passed on to the daughter cells of this particular infected cell. So that's one way that the HIV virus can propagate. It integrates into the DNA. It's not necessarily making viral particles, although there may be a low level of it. That cell replicates. It undergoes mitosis. Now you've got two cells that have viral DNA, the HIV viral DNA within it. And if that becomes activated, things can go downhill much more quickly, okay? So integrase inhibitors are another mode of treatment for HIV. The last one I want you to know are going to be entry inhibitors, okay? So if we target this step here, if that viral particle can't bind to the CD4 or the co-receptor, it can't enter the cell. If that HIV virus cannot enter the cell, it can't replicate, because it can't replicate unless it has all of that transcription translation machinery from the eukaryotic cell, okay? So in terms of that, with the HIV virus, this is an appealing level to kind of interfere with, all right? And typically when I teach face-to-face classes and I have students do an exercise, if you were to design a drug to target HIV, where would you target? Most commonly, you pick up on, well, let's not let it into the cell to start with. The other one that you pick up on, and the other way that you can interfere with viruses that usually students pick up on, is this part, the budding part. Because if you don't let that viral particle leave the cell, it can't infect another cell, okay? But there are a number of different ways to interfere with the HIV virus. And typically what they've found now is that combining a lot of these treatments are even more effective than any one individual, okay? And that's where the highly active antiviral replication therapy comes into play. That's what the HART stands for, I believe, or something along those lines, okay? Combining multiple drugs targets the virus and keeps viral load much lower. So in terms of infection with the HIV virus, the treatment option that's looking most promising is keeping viral loads low enough so that the person can live out a normal lifespan. Because if you're infected in your 20s or 30s with the HIV virus, and you can take combinations of medications that keep your viral load low so that you don't progress to the stage of AIDS, you could live out 30 or 40 years and get into a normal lifespan even though you are infected with the virus, okay? The other mode of treatment that's being looked into with HIV is developing a vaccine, okay? Now, in terms of that, viruses are really picky. Some viruses, really easy to make a vaccine to, like the polio virus. That vaccine was made way back in the 1950s when technology wasn't as advanced as it is today. We know a lot more today, but they still haven't been successful in developing a vaccine against the HIV virus, although that still is potentially possible. They haven't given up on that, okay? The last bit I want to talk to you about that's kind of unique associated with the HIV virus, I talked a little bit about needing that co-receptor in order for the virus to gain entry into the cell. There are people out in the population who have a mutation in this co-receptor. Because they've got a mutation in this co-receptor, the HIV virus can't gain entry into the cell in order to replicate, okay? So this is a great example of how a mutation can actually be beneficial in the population. Because in terms of that with the HIV virus, if you have the mutation associated with this co-receptor, you can be exposed to the HIV virus, and you can be exposed multiple times to the HIV virus, but never be infected with it, okay? Because the virus can't enter the cells, all right? So in terms of that, that kind of sums up what I want you to take away with the HIV virus, concentrating on knowing where the inhibitors are working in viral replication, okay? If you have any questions, please let me know.